give it a minute or so for people to jump on.
Sounds good.
Okay, so we're 2 minutes past the hour, so we can go ahead as people are jumping in. So welcome everyone to the IGVF Scientific Discussions, our first seminar for 2023. Happy New Year all. Today we are having one presentation by Shawn Fayer, and he's going to discuss translation of variant effect scores to evidence for clinical variant interpretation.
All righty. Thanks for the intro, Heather. Let's pull up my screen. Hopefully you can all see that okay?
Looks good. Thank you.
All right, great. Thank you. Yeah, so as Heather mentioned, I'm Shawn Fayer. I'm a genetic counselor and PhD candidate at the University of Washington, co-advised by Doug Fowler and Leah Strida in the Genome Sciences Department. And I'll be representing our center, CAVA, the Center for Actionable Variant Analysis, where we focus on variation in coding regions of the genome. And the talk that I'll be giving today is focusing on how we use the multiplex functional data that we generate in these actionable genes to resolve variants of uncertain significance from clinical genetic testing. Just looking at the chat. So today's talk is an example of how genomics technology development can significantly improve clinical genetics. And to give an example of how that's happened in the past, I'm going to just take us back in time for a moment to when I was training in clinical genetics in 2014. And at that time, if we saw a patient, oh, my slides won't advance for some reason. Oh, there we go. Okay, so at that time, if we saw a patient in cancer genetics clinic who had a personal and family history of breast and ovarian cancer, what we would do is offer sequencing of the BRCA1 and 2 genes. And with that limited test in this patient population, what we would find is that about 4% of patients had a pathogenic variant in either BRCA1 and 2. We'd call this a positive result, and it would be something that would explain their personal and family history. Another 4% of patients would have what's called a variant of uncertain significance, meaning we found a variant in either BRCA1 or 2, but we're not sure if these are disease-causing variants or just benign variation. And the remainder of patients represented in black here are patients that would have a negative result, meaning we sequence BRCA1 and 2 and don't find any variation at all explaining the cancer in the family. Sort of as our technology advanced, our sequencing technology advanced, and we identified more genes associated with hereditary breast and ovarian cancer, we began offering larger panels of genes. And then by this paper in 2019, what we found with using panels of about 40 genes, that now we're doing a better job of identifying more families that have a disease-causing variant. So now in red, we're finding about 10% of this patient population has a pathogenic variant explaining the cancer in their family. So this is great because now we're identifying more individuals. We know the risk allele in their family, so now they can communicate with their family members, and we can offer cascade testing to additional family members who may be at risk. The problem, which you can see by this chart, is now we go from about 4% VUS rate when we were only sequencing 2 genes to now about 40% of the time we find a VUS in one of these genes on one of these expanded panels. And this becomes problematic because now the majority of the patients where we're actually giving a result back to is an uncertain result. This means we tell the patient we found a variant in your gene, but we don't yet know if this is pathogenic and disease causing or just benign variation. And the best we can tell them is maybe check back with us in about a year or two, and we can see if there's been an update to the variant interpretation of the variant that we found in your gene. Now, when I say variant interpretation, what I'm referring to is when we find a sequence variant on a clinical genetic test, what we try to do is bin that variant into one of these 5 bins: benign, likely benign, uncertain significance, likely pathogenic, or pathogenic. And the way that we do this is we combine evidence from different areas from a patient's phenotype, so what brought them to the clinic in the first place, to segregation analysis. So this would be family testing where available. We'd like to see, does the variant that we found in the patient segregate in a family with disease? We'll also look at the population frequency of our variant of interest. We'll use computational predictive models, and we'll use functional assays, actually testing the function of a variant in the lab. And when we use these pieces of evidence across these different areas, we can actually weight that evidence from very strong down to supporting in either supporting a pathogenic interpretation or supporting a benign interpretation. These weights are exponentially scaled, and said a different way, if we use a piece of strong evidence for either pathogenic or benign interpretation, that's the equivalent of using 2 pieces of moderate evidence, which is the equivalent of using 4 pieces of supporting evidence. So basically, the stronger the evidence, the more predictive that piece of evidence is towards that variant interpretation. When we actually make these interpretations, we're saying something specific about that variant. So if we call a variant benign, what we're saying is that variant has a less than 5% chance of being disease-causing. When we move to likely benign, we loosen that to about 10%. So now anything between 5 and 10% chance of being disease-causing, we call likely benign. And when we, clinically, when we use likely benign or benign, we kind of use these interchangeably. Basically, anything below 10% chance of being disease-causing, we don't even report on most clinical genetic tests, and we call these benign. Conversely, anything that's 90 to 95% chance of being disease-causing, we call that likely pathogenic, and above 95% chance is pathogenic, so disease-causing. And what you might notice is the biggest chunk here are the variants of uncertain significance. So anything that has a between 10 and 90% chance of being disease-causing, we kind of throw our hands up. We don't know yet if these are disease-causing or still just population variation, and we call these uncertain significance. You might be able to predict that sort of the evidence that I showed you on the previous slide, it's really hard to come by all of those pieces of evidence for any given variant. And since the biggest piece of the pie is this uncertain significance, if we go to the ClinVar database and look at all the missense variant interpretations, what we'll find is that the majority, just over 80% of missense variants in the ClinVar database that have been clinically annotated are variants of uncertain significance. And this is a problem that's actually been growing over time. So if you did the same ClinVar data pull annually from 2014, what you'll see is there's this rapid expansion of the variants of uncertain significance and a more modest increase of the more definitive interpretation categories. And the reason for this is actually pretty simple. We're just sequencing more genes, and the more genes that we sequence, the more rare variation we identify. And the more rare a variant is, the less evidence that we have to interpret that variant, so it's more likely to fall into this VUS category. Now, while we can't do very much about the rarity of these variants, what we can do is generate functional data at scale to help resolve this VUS problem. And so what we do in Kava is we generate multiplex functional assays. And a very simple view of what this looks like is we'll take a cell line of interest and we'll identify a gene that we want to functionally annotate. And so in that gene, we will introduce a single variant to each one of these cells such that each cell has a different variant of that gene. And then we'll throw these cells in pool through a functional selection where if a variant is pathogenic or disease causing in this gene of interest, it might cause them to be depleted in this pooled population of cells, and benign variants can continue to proliferate and will become overrepresented. So we can use that functional selection to then sequence the variants in this pool of cells and generate a function score for each variant. And what we'll be left with is a map that looks something like this. Function scores for each variant that we measure in the assay. And what we would like to do when we generate these maps is integrate this into clinical genetic testing to help resolve a lot of those variants of uncertain significance. Now, when I joined the Fowler and Starita labs, there had been a handful of these multiplex functional assays that were published, but there hadn't been sort of a systematic analysis of how many variants from clinical genetic testing, or how many VUS from clinical genetic testing can you resolve with this data. So we just asked a simple question, what is the clinical utility of this multiplex functional data and reclassifying variants of uncertain significance? And so to answer this question, we took some of those already published multiplex functional data sets across 3 different tumor suppressor genes, BRCA1, TP53, and PTEN. And I'll touch on each of the assays individually. So starting with BRCA1, we took the saturation genome editing data that was published by Greg Finley from the Shenjuri lab here at the University of Washington. And what I'm showing you here is the function scores for variants of known clinical impact that were measured in the SGE experiment. So along the x-axis, we have our function scores, and colored in red and orange are the pathogenic and likely pathogenic variants measured in the assay that are from ClinVar. And the blue and light blue are the benign and likely benign ClinVar variants that were measured in the assay. And so you can see this assay cleanly separates the known pathogenics and benigns. Next, for TP53, we had available to us 4 different previously published multiplex functional assays, 2 that looked at any loss of function variants, and 2 that specifically screened for dominant negative variants in cell lines that were on a background with wild type TP53. And then finally, we had 2 different assays for PTEN, one in mammalian cells that looked at protein abundance, and one in a humanized yeast strain that looked at the phosphatase activity of PTEN variants. So across these 3 genes, we have thousands of variant effects measured. And what we wanted to do is now integrate this with clinical variant interpretation. So the way that we did that is we collaborated with Ambry Genetics, and they graciously sent us their interpretations across the 3 genes based on testing that was sent to them from patients who either had the phenotype or family history associated with BRCA1, TP53, and PTEN mutations. And what you can see in this clinical data set is that, much like the ClinVar data I showed you earlier, the majority class of variants that Ambry is interpreting are also variants of uncertain significance. So we integrated the multiplex functional data to try to reduce that VUS piece of the pie here. And the way that we do this, we can largely break down into 3 different steps. First, we're collecting and combining the multiplex functional data. Then we're calculating that strength of evidence that I mentioned earlier based on the assay's ability to distinguish between known pathogenic and known benign variants. And then we take that strength of evidence and we use that to apply the functional data with the clinical variant interpretations. So I'll first walk through the 3 steps with the BRCA1 data, since it's the sort of the simplest case, we just have one assay. So for step 1, essentially one of the first things you want to do is just a sanity check. So if you have a multiplex functional data set, you want to ask, is the data set telling you what you think it's telling you? So for BRCA1, a way that we can do this is by plotting the nonsense and the synonymous variants that are measured in the assay. Because loss of function is a known mechanism for BRCA1 pathogenesis, we should be able to separate the nonsense variants from synonymous if the assay is actually telling us something meaningful clinically about BRCA1 mutations. So here you can see we clearly have separation between the nonsense variants in this purple-pink color and the synonymous variants in this green color. The vertical lines represent the 95% cutoffs of the bimodal distribution of all the data in the assay. And what's nice is the nonsense variants mostly are distributed to the left side of the dashed purple line. So anything now that falls into this purple shaded region, we're calling functionally abnormally in the assay because they behave like the nonsense variants. And similarly, anything that falls to the right side of the dashed green line in this shaded green area, we're going to call functionally normal because these behave like the synonymous variants in the assay. So the next step is to now calculate that strength of evidence. And the way we do this is through a statistic that we call the odds of pathogenicity. And so to illustrate how we do this, I'm now showing you, again, the distribution of variants in the assay. But this time, these are the variants in ClinVar that are either known pathogenic or likely pathogenic in red and orange, or known benign and likely benign in blue and light blue. And so first, we'll start with the functionally abnormal side of the distribution, so to the left of the leftmost dashed line. And when we calculate the odds path, what we do is we ask how many of all of the variants on the left side, what proportion of those are known pathogenic variants. And we can, through a Bayesian framework, calculate this odds of pathogenicity or odds path, which in this case comes out to 52.4. And 52.4 actually corresponds to strong evidence for a pathogenic interpretation of any variant that now falls to the left side of the left vertical dashed line. On the other side of the distribution, for variants that we're now calling functionally normal, we also calculate an odds path based on the proportion of true pathogenic variants on that side of the distribution. And this calculates out to 0.02, which is now also strong evidence, but in this case for a benign interpretation for any variant that falls into the functionally normal side of the distribution. So we now take these evidence strength codes and look for Ambry's variants of uncertain significance to see which ones fall into which sides of the distribution, and apply those evidence strength codes to Ambry's clinical interpretations, combined with the evidence that Ambry had already used. And what we find is that 49% of those VUS were able to be reclassified using the multiplex functional data from the SGE experiment. So what that breaks down to are 49 of the VUS and Ambry's data set are now moved to likely benign, and 5 VUS are now moved to likely pathogenic. And I just want to highlight at this point that each one of these VUS represents a patient, a group of patients, or even a group of families that had a VUS from clinical genetic testing. They couldn't really do anything with that information, but Ambry then sent them an updated sequencing report telling them that either their variant has been moved to likely benign, meaning it's not thought that there is a variant in BRCA1 that is explaining the cancer in the family, or moved to likely pathogenic, which opens up additional screening and surveillance to these individuals, as well as cascade testing to other family members who might be at risk of having the variant, who might want to follow up with enhanced screening, surveillance, and risk reduction options. So this is the case for BRCA1. We reclassify nearly 50% of the VUS using the multiplex functional data. But now when we move on to TP53, things get a bit more complicated. And the reason for that is now we have 4 multiplex functional assays instead of just 1. And if you look at these distributions and in comparison to that BRCA1 data, we actually don't have the same level of separation between known pathogenic and known benign variants. So if I were to take any one of these assays and calculate the odds path, I'd be limited to supporting evidence, which would then limit the impact of this data in clinical variant interpretation. However, we reasoned that because we have these 4 different assays, which are either being performed in different cell lines or have different functional outputs, we could combine these assays with a probabilistic classifier to come up with a unified prediction of the variant effects. And this was at the IGVF meeting, actually a theme that I saw come up a few times, making multiple measurements per variant with different assays. So this is a nice demonstration of when you have such a combination of data sets, what you can actually do with the data clinically. So in order to do this, we trained a Naïve Bayes classifier. And this classifier was trained on all the clinical variants of known effect that I'm showing on the screen here. And in our cross-validation of this classifier, 39 of the variants were predicted to be functionally normal, and 123 were predicted to be functionally abnormal. When you overlay the actual interpretations of these variants, what we find is that all of the 123 variants that were predicted to be functionally abnormal by the classifier were true pathogenic variants, and 32 of the variants predicted to be functionally normal were benign, with 7 misclassified pathogenic variants as functionally normal. We can now calculate our odds path based on the classifier predictions. And for variants that are predicted to be functionally abnormal by this classifier, we can apply strong evidence because the classifier does a good job of predicting this class of variant. And then for the variants predicted to be functionally normal, we can give moderate evidence because it doesn't do quite as good a job at predicting benign variants. When we then took these evidence strength codes and integrated them with Ambry's interpretations, we were able to reclassify nearly 70% of the VUS in the data set. And so this is a really exciting result for me because we go from a set of functional assays where individually we can only hit supporting evidence, but when we combine the 4 of them together, we actually get decently strong predictions for pathogenic and benign variants. And then when we combine that with the true clinical data available to us in the Ambry data set, we reclassify nearly 70% of those VUS, significantly reducing the amount of VUS for TP53. And then finally for P10, because again, we had multiple assays, we were hoping to sort of do a similar assay, use a similar classifier for P10. The problem with that, though, is, at least at the time of the publication and the last time I checked ClinVar, there were only 2 known likely benign variants, missense variants in P10. And for that reason, we're very limited in the odds path calculation for these assays individually, but also it limits our ability to train a classifier to unify the 2 data sets. So for that reason, in this analysis, we actually took a step back and reflexed to the P10 variant curation expert panel guidelines for variant curation. And we're still able to use these 2 functional data sets because they're specifically mentioned in those guidelines. And we were able to reclassify 15% of the P10 VUS, moving 7 variants from VUS to likely pathogenic. So I hope, now sort of looking back at all 3 genes, that I've convinced you that there's actually quite a high degree of clinical utility to multiplex functional data, especially when generated in clinically actionable genes, like these 3 tumor suppressor genes. And we're actually really excited in Kava because we're now generating additional multiplex functional data sets across many more known clinically actionable genes. At the top of both of these lists, PALB2 and factor 9 are the ones that are closest to being integrated into clinical interpretations to help close the gap for these 2 genes. But many more genes are being measured in both the saturation genome editing assay as well as protein abundance. And so with that, I would just like to thank both the Fowler and Streeter Labs, especially Doug and Leah, for their shared enthusiasm for variant interpretation and integrating these assays into what we actually do in the clinic. A big thank you to Ambry Genetics for sharing their data, which really made this analysis possible, as well as our funding sources. And yeah, I'd be happy to take any questions.